I'm happy to be here for episode 4 of the NEATCast, a podcast about mental health advances applicable to our daily lives. Today, I'm happy to be joined and to invite Dr. Scott Wilke, Assistant Professor of Psychiatry at University of California, Los Angeles, and recently appointed to be the Penske Family Chair of Neuromodulation. This article that it was recently released January 2025 is a preprint, and I'm sure will be published at some point in time as research goes. The article is called "A Cell Type Specific Mechanism Driving the Rapid Antidepressant Effects of Transcranial Magnetic Stimulation." I've known Scott for a while and have seen this story evolve from a few years back when he was first starting to get it up and running till now. And I'm very excited to talk with him about it now that the paper has been released. As always, you can find our podcast on multiple places like Apple Podcasts, Spotify, and YouTube. All right, Scott. So let's start off this conversation by having you just tell us a little bit more about what transcranial magnetic stimulation is and why it's important for psychiatrists and psychiatry.
Yeah, sure. Well, first of all, I just want to say thanks for having me on the podcast, Dhakshin. We've known each other, I think, now for probably 20 years.
Something like that.
And it's just really exciting to see what you guys are doing. I think a podcast about neuroscience and psychiatry is such a cool idea. So really excited to be on. And your question was about what is transcranial magnetic stimulation? People call this TMS. TMS obviously stands for transcranial magnetic stimulation. And basically, TMS, as you know, is a way to non-invasively stimulate parts of the brain in people. So you're taking an electromagnet, you're placing it against the side of the patient's skull, and you can actually stimulate the part of the brain that's directly underneath the magnets. This is using pulsed electromagnetic fields, and those fields drive activity in the neurons, the cells that are in the brain region that you're trying to target. And it's important for psychiatrists because it's become a treatment that is quite effective for depression and other psychiatric disorders. So it sounds relatively new and technically novel, but it's actually been around now for 10, 15 years in use in the clinic. And it's really helpful for us as psychiatrists because it's a treatment that we can use for people who haven't responded to medications, antidepressant medications, and psychotherapy, other more kind of typical treatments. I just wanted to highlight one thing that I think is really interesting about TMS, or maybe an advantage of TMS, because it's actually able to focally target brain regions that might be involved in depression, and that has some advantages. In particular, when you treat someone with a medication, the medication goes everywhere in the brain and everywhere in the body, and so it ends up affecting physiological processes that are not the ones you want to target, and that's one of the reasons people have a lot of side effects from medications and other treatments sometimes. And so, as you know, no treatment is free of side effects. There's certainly side effects when people are treated with TMS, but the side effects tend to be pretty manageable with TMS, and most people tolerate it really well. So that's, one of the reasons I think it's such a great treatment. I like to use it for patients who are depressed.
That's awesome. That's just such a great explanation for what TMS is and why we use it. I heard a few different things. I just want to reinforce that you were saying one of the advantages of TMS compared to other medications is that it can target maybe brain circuits in a more specific way. So medications, you take a pill and it's basically affecting a neurotransmitter system everywhere in the brain, right? And not even in the brain, everywhere in the body. And we have a bunch of neurotransmitter systems doing all kinds of things in our body. And with TMS,
Exactly.
really targeting a particular circuit, and that's kind of one of the maybe the advantages of it. So what has been done? Like if people asked you, how does TMS work to help treat depression, right? This is a common question people might ask us in the clinic if we're recommending it. Um, what would you say?
Yeah, well, that's, it's a good question, and it's one that people have a lot. The truth is that there's probably way more things we don't understand about how TMS works than we do understand. This is also the case with medications, I think, but I'm always really transparent with people about the fact that there are a lot of unknowns in terms of exactly how the treatment works. We know that TMS is activating the cells and the circuits in the brain region that you target. And one of the important things about how we deliver TMS as a clinical treatment is that we deliver these pulses in specific repetitive patterns. So you'll often hear TMS as a treatment referred to as repetitive transcranial magnetic stimulation, or rTMS. And that just means that instead of just, stimulating the brain once, you're actually stimulating it repetitively in a specific pattern. And so those patterns can be slow patterns, they can be fast patterns, they can be bursting patterns. And the general idea is that the specific pattern causes some changes in the brain that people broadly refer to as neuroplasticity, which just means that we're sort of changing the activity of the neurons. We're actually causing some structural and functional changes in the brain that we think of as being beneficial. And typically, what you're doing is you're targeting TMS to a brain region that you think is either under or overactive in people with a particular condition. So, for example, in depression, it tends to be the case that people have reduced activity in a region called the dorsolateral prefrontal cortex. And by delivering specific patterns, we think that you can sort of amp up activity in that region, or in other cases, you could maybe even dampen activity if you think that the region is overactive. So we know that these particular TMS patterns are causing sort of lasting changes in the activity of these brain regions in people, but what we really don't know is exactly how that, the mechanics of how that happens. And this is the question that I've been really interested in is, what is the actual mechanism? What are the, specific cell types and circuits? What are the changes in the brain that actually are responsible for producing an effect that alleviates depression for people?
Mm-hmm. This gets into the work that you did, which was in a mouse model. You had to develop a mouse model of TMS, and I know from talking with you that that was not easy to do that. Before we get into that, though, I want to just ask you a question. Why did you even need a mouse model? Like, why not just study synaptic plasticity and all of this stuff in humans?
Yeah, a great question. I mean, the answer is that when we try to study, it's very useful to study things in people and patients directly, and many people have done that, and there are many important insights that have been gleaned from that. But when you study mechanisms in people, you're just fundamentally limited. You can't get inside the head. You can't see the specific, synapses or circuits or cells and how they're changing. You're sort of limited to techniques that are non-invasive. No one's going to agree to have their head cut open and their brain looked at to figure out how TMS works. So having an animal model allows us to like look at the what's happening with TMS treatment at a level that you just wouldn't be able to in people. And so in that way, it's sort of a complementary approach. So we can learn some things in animal models, and there will be caveats to those things because mice, uh, rats, things like this are not the same as people. But we can still discover sort of fundamental mechanisms that might influence how we think about how TMS works as a treatment.
Yeah, great. Okay, so tell us what you had to do to take a human TMS device, which, for our audience, these are pretty big. Like the size of a coil that we use in humans is, maybe about the size of like five mice or something like that, 5 or 8 mice. So how
Yeah.
did you develop a mouse TMS coil, and what were the challenges?
Yeah, so there's a lot of challenges. And this is the point where I want to briefly pause and give credit to the rest of the team that worked on the project. We often talk about the science in our labs as we did this, we did that, but it's really only possible because of the hard work and technical prowess of the students, the postdocs, the scientists in the labs that are doing the work. And in this particular case, the work was led by a really talented MD PhD student at UCLA. His name is Michael Gongwer. And very important for me to say that, this is a group effort, but Michael led a lot of the really technical aspects of this and made some of the key discoveries. We also had funding for the work, which is really important. You can't do good science without money to support it. So this is dependent on funding from the NIMH, and also from the Penske family. So with that said, your question is about how did we do this? How hard was it? As you mentioned, TMS coils are big. I often describe the TMS coil as maybe like the size of a brick. And the main challenge here is how do you scale that down to the size of a mouse brain if you really want the model of TMS to be similar to what we do in people, then it has to kind of maintain this size or focal feature of the treatment, right? And for people who don't have a sense of scale, the size of a mouse brain is maybe the size of like the tip of my pinky finger. So it's pretty small. Um, so it turns out you can't, it's not so easy to just shrink these things down. There are a lot of like physics and other technological issues that arise with that. And this is the thing that's really sort of stymied the development of animal models of TMS for
Hmm.
a long time.
Hmm.
Um, and if you look at what people have done, which is some great work, obviously, they've either used really large coils that are probably so big that they stimulate most of the mouse brain, if not the entire mouse brain.
Hmm.
Or in other cases, they've used these really miniaturized TMS devices that are not strong enough to actually drive firing in the neurons in the brain. And so we wanted to have a way to sort of have both of those features, focal stimulation and strong stimulation, the way we do in people. And so again, we were dependent on our collaborators to do this. there's a group at the National Institute on Drug Abuse. This is led by scientist Hanbing Liu, and he developed what we think is the first rodent TMS coil that's capable of both of those things. So it's able to stimulate a subregion of the mouse brain, and it can do it strongly enough to actually cause the neurons to fire the way we think they do in people. Um, so that's like the first level of challenge, right? Is just developing this coil. And
Mhm.
Han Bing was able to develop this amazing device that can do that. And so we, as we do in a lot of science, we collaborated with him, and he's sent us a couple of his coils, which we used. That only got us so far, though, because you still have to come up with a way of treating mice the way you treat people. And the the goal throughout this entire project was to do this in a way that was as similar as possible to how we treat patients in the clinic. And if you do it in a different way, the results may not be as relevant to the clinical uh, approach, right? So we did a few things that I think are interesting to achieve that. We wanted the mice to be awake, so we developed a way to fix their head in place, and then they actually walk on the back of a running wheel. And so their head is still, but they aren't restrained in a sort
Mhm.
of stressful, unnatural way. So they can just kind of walk on this wheel. Um, One of the biggest challenges to doing this is that when you shrink the coils down like this, they can really easily overheat.
Mhm.
And so the whole thing can melt down if you're not careful. So, one of the biggest, I mean, if you can imagine, just keeping the thing cool was one of the biggest challenges. And when we first started doing these experiments, we would pack the whole thing in dry ice,
Mhm.
and we'd be constantly smashing the dry ice and trying to keep this thing at a low temperature. And Michael Gongor, who's the MD PhD student I mentioned, he came up with a really elegant solution for this. He actually worked with his dad, who's in heating, ventilation, and air conditioning. And
I was just
it's
gonna
sort of
say, it
one of
sounds like you need
these,
a AC engineer, basically.
yeah, and that's basically what we got. And Michael worked with his dad. They developed this amazing heat sink device, which is actually a liquid cooled copper device that encases the coil and just basically sucks the heat out of it really efficiently. And that's actually, it sounds very technical and logistical, but without that, none of these experiments are really possible. So that's one of the problems that we had to solve.
Yeah. And you did a lot of nice validation. I think the first part of your figures in your paper was really validating that, hey, this coil works. You have some antidepressant effects, right? I'm going to skip over that question for now because I do want you to talk a little bit more about the antidepressant effects and what that means. But I want you to dive in. So, like, what did you find? You developed this. You took a coil that other folks had. You modified it in order to be able to do what you wanted to do, and you really thought a lot and were very careful about how do you make the results as translationally relevant, as likely as possible that anything you find may be similar to what's going on in human TMS, right? So tell us what you found.
Yeah, so I mean, as you mentioned, the first thing we found is that we can have effects on the mice that are similar to what we see in people. And I won't go too much into the details here, but basically what we did is in all of our experiments, what we do is we model chronic stress in the mice. And when mice are exposed to a chronically stressful environment or experience, they develop features that are similar to human depression in some ways. And so the first thing we showed is that if we deliver a clinical treatment protocol, the same protocols that we do in people in our clinic, then we could actually reverse some of these depression-like changes in behavior in the mice. Okay. So, but ultimately, this, like you said, this is just validation. So what we want to do is we want to really actually understand what's happening in the brain. And people think of brain regions as having like sort of specific functions, like this region does this region does that. But I think sometimes people don't appreciate just how complex these brain regions are. So individual brain regions like the prefrontal cortex could have over 1 billion neurons, and there's just an enormous amount of complexity in there. So the neurons are not all the same. There are many different types of neurons, and those neurons have different functions, different properties, and they're connected with each other in different types of circuits that are actually the functional units of the brain. And so we wondered, does TMS treatment just sort of globally affect all the neurons in the prefrontal cortex, or does it have more specific effects on distinct cell types? And that was sort of like the hypothesis that we had, is maybe there are distinct effects on distinct cell types. And so we decided to look at two sort of classes of neurons in the prefrontal cortex. There's many classes, there's subclasses. We looked at two broad classes, and they have really long names. One is called the intratelocephalic neuron, which we call IT neurons. The other is called the pyramidal tract neuron, which is called PT neurons. And the name just basically means that each of those types of neurons is connected to different parts of the brain. And we thought, well, maybe what we'll find is that the effects of TMS are different in these two types of neurons, and that's exactly what we found. Do you want me to go into some of those findings?
Yeah, well, I'm curious because part of this is also thinking about, how you came up with that hypothesis. So I'm gonna give you my my view, which is as a scientist, I don't know if I would have had that hypothesis, which is what I found so incredibly interesting about your work. I would have thought more like your straw man hypothesis is probably more what I would have thought is, hey, TMS is an electromagnetic thing. It's affecting the biophysics of these neurons. And to the degree to which it's doing that, neurons will respond accordingly. But you found that the way that neurons are connected within a network affect how they respond to brain stimulation. Now, this is, in my opinion, super unexpected, and you showed this very, very clearly and robustly. So how did you come up with that idea that, hey, there may be something different about these two classes, right, that might predict whether they respond to brain stimulation?
Yeah, I mean, so I guess to explain that, I think you have to sort of go back to this idea that there are different types of TMS. It's not all the same, right? And so clinically, what we observe is, there are all these different ways of doing the TMS, high frequencies, low frequencies, bursting frequencies. And, what you often find is that different people respond differently to one or the other of these sort of treatment approaches. And so I sort of had the question of like, why would, like, if these are stimulating just like all the neurons, why would you see that the different people respond differently? And maybe it's because one person has dysfunction in a specific circuit, cell type, or network, and if you find the right TMS treatment, you can sort of modulate that network. So it's a bit based on that, and then it was sort of like a guess. You
Mm-hmm.
know, there was not there was nothing special about the IT and PT neurons other than that these are pretty different. We thought these are pretty different. They do different things. They're connected in different networks, and maybe uh, you know, TMS would affect them differently. The only other thing I can say is that there is some clinical data that suggests that connections between different prefrontal, different cortical regions are important for the effects of TMS. And
Mm-hmm.
the IT neurons are actually the neurons that mediate those types of connections. So
Mm-hmm. Mm-hmm. Cool.
that's how we
Yeah.
came up with the hypothesis.
Yeah. So partially based on your clinical experience, which is invaluable, partially based on kind of thinking about from a network level, hey, if this is how we think maybe human TMS works, this would be the class of neurons that might mediate those effects in mice. And partially with a lot of science, it's like, well, we're not going to know unless we test it out. So you go with your best guess, you test it out, and sometimes you're right and sometimes you're not right, but either way, you learn something. Um,
Exactly.
Cool, cool. Okay, yeah. So tell us a little bit more about like what did you find that TMS was doing to these? You basically found that you study these two types of neurons. You found TMS mostly seemed to affect the IT neurons, this one particular class of neurons.
Mhm.
What did you find that TMS was doing to these neurons that seemed to mediate the antidepressant effects?
Yeah, so there's two ways we looked at the neurons. One was sort of a structural way, and one was a functional way. And the first thing we found with the vision is let's measure the activity of these two types of neurons, both during the TMS treatment itself and afterwards during these depression-like behaviors. And what we found was that the IT neurons exhibited these increases in their activity as a consequence of the treatment. And this includes like during the behaviors that we think are related to depression and which were improved by the treatment. There's actually an increased engagement of the IT neurons as compared to the PT neurons. So that was really interesting and a clue as to what might be going on. We also tried to go in and look at the structure of the two types of neurons and how that changed in response to TMS treatment. Um, it's been known for some time that when you expose animals to chronic stress, one of the things that happens is there's sort of an atrophy of the connectivity of neurons in the prefrontal cortex. So they will lose these sites of communication called synapses. And so the neurons seem like they are struggling to communicate with each other as well as they would normally uh, do. And this affects both types of neurons. They both lose these structures that are the sites of synaptic communication. But the TMS treatment, we looked at these two neurons, actually restored these synaptic structures on the IT neurons, but not the PT neurons. So what we saw are both functional correlates and structural correlates of TMS that were impacting this specific cell type, and presumably the circuits and networks that cell type mediates.
Mm-hmm. One of the things that, as a psychiatrist who recommends TMS to people, one of the things people always ask is, how does TMS work? And I think there's tremendous value in being able to say, what you just said to people, that hey, we now have very good evidence that TMS works by activating neurons that we think are involved in depression and not properly activated. and promoting new connections, new synapses. And I think people understand that, and they I think they're more likely to get a treatment if they think we know how it works, as opposed to us saying, we don't know how it works, but it works, you know. So
Yeah.
the clinical evidence is helpful. You need the clinical evidence. Oh, something works. But if you don't know how it works, it doesn't feel great to recommend it, and I think patients themselves are a little bit like, what kind of field is it? So I think there's huge value in just that idea that we're starting to understand how these treatments work better and being able to communicate that. But of course, I'm always excited to say and to ask you, do you think that there could be some new treatment approach based on these findings? Do you think that this model system would allow you to develop maybe new stimulation approaches that could target the PT neurons or different classes of neurons? Like, where do you see this going forward in terms of next steps for you?
Yeah, great question. Super important. You know, I think one of the things is we don't really know what this IT versus PT thing means. Is that a feature of TMS treatment, or is it a bug, so to speak, right? So is it good that TMS is only
Right.
having this effect on the IT neurons? Is that is that, or would it be better if we could get the TMS to work in a way that modulated both of the neurons
Right.
types?
Right. Yeah.
And I don't think we have a great answer to that question yet. Although I can tell you that there are other types of antidepressant manipulations that seem to affect the PT neurons and depend on the PT neurons. So it's not like only IT neurons and their circuits are important for depression. Probably both of these are important for depression. And what we want to do next is sort of understand, when you change the way TMS is delivered, can you target different types of neurons and get different types of effects, right? So I think of it like almost like tuning a radio, right? And the way we do TMS now, we've sort of picked a few frequencies on the radio dial, and we know that they can be helpful. But there's probably 99% of the possible places on the dial that we haven't tested yet. And if we could understand how using different frequencies or patterns of stimulation could be targeted to specific cell types or circuits, then that opens like an entirely new set of possibilities in terms of personalizing the treatment to the problems that specific people are having, better targeting the problems that arise in different types of disorders, things like that. So this is kind of the next thing that I think we're going to pursue, is trying to see if different types of patterns actually can target different types of neurons. And maybe what you need to do is uh, you know, do TMS in a way that you can activate both the IT and the PT neurons, or
Mhm.
maybe sometimes you want to modulate the PT neurons, sometimes you want to modulate the IT neurons. So I think if we can figure that out a little bit, we have the potential to translate some of this into, better or more precise treatments for people.
That's super exciting. And I so agree with that. I think that is the next step. Um, in the field of neuromodulation, it is expensive, difficult, and time consuming to do parameter testing in this way, and impossible to do it at the level of specificity you're talking about, right? Different cell
Right.
types, different circuits, different networks. So, I mean, again, it speaks to the role of a good translational model in order to answer some of these questions that can really inform improvements in treatments. Okay, last question for you, which is, this isn't a good journal club unless we tear apart or highlight all of the flaws of the research or the paper. So, tell me what you think are some of the limitations of your work, and is this a limitation that could be addressed in the future? Or is it just kind of a fundamental limitation of animal research that, we have to accept, but think about, there are limitations of every type of research, like you were saying.
Yeah, I mean, I think there's always going to be limitations when you're trying to study psychiatric disorders in animals, and we sort of hinted at that, earlier in our conversation. I think there's both technical and sort of conceptual limitations, right? So like one of the issues here is that the way the mouse brain is organized is pretty similar to how a person's brain is organized. Like we have all these similar regions and they tend to be connected in similar ways. People and mice both have these IT and PT neurons. It's a very highly conserved feature across species and across cortical brain regions. But at the same time, there are really important differences between how the mouse brain is structured, how it functions, and how the human brain is structured and functions. And I think you never that's why you do the translational work, because you
Hmm.
don't know for sure how these findings would hold up in people. So there's a lot left to do in that regard. um and the behaviors that we're modifying, I mean, we sort of hinted at this earlier as well. We're always really careful not to say that our mice have depression,
Hmm.
or that we are treating depression in our mice, because depression is a human condition, and certainly mice don't have depression in the same way that people do. So I always say that the behaviors are depression related or depression
Hmm.
like,
Hmm.
and we say the effects are antidepressant like, because we want to be really careful about making strong claims that are not justified. And so you never know how those things are going to translate, which is again why it's so critical that we are doing the treatments the way we do them in people, because that's
Hmm.
really the thing that assures that we are doing something that's clinically relevant, and our results might be valuable. But
Hmm.
ultimately, I think, you need to take these things and test them in people, and that will really tell us, to the extent to which we've discovered something that's meaningful and useful.
Scott, thank you so much. It was great having you on, and yeah, I look forward to seeing what comes next from you.
Yeah, thank you so much for having me. Appreciate the opportunity, and excited about what you guys are doing with the podcast.